Yep. And then at the end, and then we go up, we come up and save it appropriately. It's all you now.
All right.
All right, it's 4 o'clock. My understanding this is before we end the spring semester seminar series, any announcements? I realized that I've been charged. All right, we'll see what we can do. Okay, any other announcements? Okay, so if not, I would, it's my pleasure to announce today's speaker in the Angela. Today she's got, initially we were planning on to have this today she's still and the things that she's going to but she's going to talk about
Thank you for the introduction. I appreciate you all coming today. Like Dr. Shim said, I had the opportunity to cancel this presentation, but I thought that this would be a really good opportunity for me to not only share my research, but to also practice in presenting and talking about what it is that I do on a daily basis and over the past 2 years. And so the main project that I'll be talking about today is on the characterization of secondary metabolism regulation in Fusarium verticillioides using subnetwork module prediction. So just a presentation outline. I'm first going to go over this introductory study, which is kind of an appetizer, I guess. And it's on the investigation of etiology of Pokkah Boeng disease of sugarcane in southern China. And so this is work, as Dr. Shim mentioned, that I had done at the Fujian Agriculture and Forestry University over the summer of 2014. I was there for 13 weeks total. And it was an interesting experience, and so I'm excited to share it with you guys today. And then my main project that I'll be talking about is, of course, the characterization of secondary metabolism regulation in Fusarium verticillioides using subnetwork module prediction. And so just a quick background of my appetizer study, or quick overview of what I'll be talking about. I'm going to go over the background as well as the methods and results that I got and a conclusion. If you look at this image on the right, this is the classic symptomology that we see in mature sugarcane plants that are infected with this Pokkah Boeng disease. You can see this malformed top. We call that knife cut. And it's often what we would see out in the field of infected plants. So just a quick background, sugarcane is a perennial true grass of the genus Saccharum. It's a row crop. It's high in cellulitic material. And in 2012 and 2013, China produced approximately 13.1 million metric tons of sugarcane, making them the third largest producer of sugarcane in the world. And in southern China and within the Southeast Asian region, smut, stalk rot being Pokkah Boeng, and mosaic virus account for the majority of the disease. This is me here with Dr. Lin. He's a sugarcane breeder there at Fafu. I had the opportunity to meet with him and kind of interview with him and find out a lot about the problem with the disease of sugarcane in the region and what he's seen throughout his experience as a breeder. So Pokkah Boeng disease is an emerging disease of sugarcane which has been causing major yield losses in most sugarcane producing regions around the world. It's predominant in Southeast Asia. It has made a presence here in the United States as well as the Latin American region. However, it is not responsible for causing major yield losses like what has been seen in Southeast Asia. And like I had said, the typical symptoms include these malformed leaves and stalk rot. It also includes red streaking and chlorosis. And the causal agent of this disease is recorded as today as Fusarium moniliforme. Now, over the past decade, Fusarium moniliforme has been somewhat broken down into many different species, and so ultimately researchers have yet to come to a consensus on which particular Fusarium species is the primary causal agent for Pokkah Boeng disease of sugarcane. And so through this project, I wanted to be able to identify what that primary causal agent was within southern China. So how did I do this? I went out to the field, I did some sampling for disease plants, I isolated the fungi. I then did a dinitrosalicylic acid-based enzyme assay to detect for both mono and disaccharide sugars in the solution. I did ITS sequencing of the fungi that I had collected, as well as calmodulin and elongation factor sequencing for the putative causal agent. I also did some growth assays and some pathogenicity assays to follow Koch's postulates, and then some microscopy. And you can see this is me here. I'm there at the lab in Fafu. Doesn't look much different than the labs here in the US. So in this image, I'm actually sterilizing some of the subsamples that I had taken from the field. So this is the sugarcane field that I was sampling from. It was approximately 3.5 hectares. I split it into quadrants in order to help me establish a systematic random sampling approach. The primary cultivar grown in this field is a hybrid known as ROC22, and is a hybrid of Saccharum officinarum and Saccharum spontaneum. And it's actually somewhat of a monoculture. They grow this cultivar throughout southern Asia, as well as other sugarcane growing regions. And a total of 74 diseased plants were collected from the field. And these are some of the samples that I brought back. These are some really good examples of what I was looking for when I was in the field. This is obviously from mature plants. You can see the malformed leaves, this leaf cut symptomology I was talking about before in a previous photo. In this bottom right and top left corner, you can see the typical symptomology of stalk rot. And in the bottom right corner, this is what we often see in younger sugarcane plants, what we call this leaf curling symptomology. And so I was a little bit lost, I'll admit. I was pretty green going out there, a new graduate student in a foreign place, and I didn't really know where to go from here. And I was really lucky that Dr. Jim Starr had recently retired and was teaching a nematology course there at Fafu, and so he gave me some really good advice about how to properly take subsamples and to isolate the fungi that I was looking for. And so what he recommended was that I cut out subsamples lateral to disease symptoms And the reasoning behind this was that these lesions would likely have secondary infectors as well as saprophytes, and so it would be better to sample in lateral to those lesions. So after I cut out these subsamples, approximately 1 centimeter squared, I sterilized with 10% bleach and 70% ethanol. And then I inoculated onto a complex media with antibiotics in order to inhibit bacterial growth. I decided to incubate the subsamples at 25 degrees Celsius. So this is approximately the ambient temperature there in southern China at the time. Obviously, it heats up a little bit more later in the summer. And then once I saw fungal growth, I transferred those fungi onto separate plates. And I was able to isolate a total of 137 different fungal strains. And these two pictures don't really do it justice. I had a ton of plates. I didn't know what to do. I was a little overwhelmed, but it was very interesting. I mean, we had so much diversity. There was many different types. You can see that we had to composite some of the samples because I was so overwhelmed. I didn't really know what to do. But then we came up with a hypothesis that based on the fact that sugarcane is high in cellulitic content, the causal agent for disease would be able to produce a high level of cellulase. And so I performed this dinitrosalicylic acid based assay in order to narrow it down to the top 10 highest cellulase producers out of the 137 strains that I isolated. Now what's really funny is that strain 20, as you'll see in later slides, is the putative causal agent for the disease, but it wasn't the highest producer of cellulase. In fact, strain 47, strain 22, and strain 37 were the highest producers of cellulase of all the fungi that I isolated. So at the time, Dr. Shim was working in South Korea, and so I extracted DNA, sent it off to him, and he performed ITS sequencing there. With that, he was able to identify strain 22 as Alternaria, strain 27 as Phomopsis, strain 37 as Phoma, and strain 47 as Epicocum. We knew that strain 20 was a Fusarium strain, but I wanted to go ahead and do some comodulin and elongation factor sequencing to further validate that. But unfortunately, I was still getting a little bit of variation, and so I decided to go ahead and do some microscopy. And paying close attention to this Fusarium laboratory manual that is written by Leslie and Summerell in 2006, I was able to identify the fungus based on morphological characteristics. So Fusarium proliferatum falls into the same mating class as Fusarium fujicroi, although what makes it distinct from other closely related species is that it's able to produce both monophyllide and polyphyllide conidiaphore structures. And so we proposed that the fungi that I was able to isolate is a Fusarium proliferatum. So the next thing I wanted to look at was the growth rate of the fungi. I decided to stick with my Fusarium proliferatum strain as well as Phomopsis and Phoma, which are some other known pathogens. And we proposed that the causal agent for disease would be more resistant to cell wall stress. And so H2O2, Congo red, and SDS were used to mimic that environmental stressor. And what I found was that Fusarium proliferatum didn't grow significantly faster or slower on complete media or complex media with H2O2 or complex media with Congo red. However, it did grow significantly faster on complex media with SDS. In addition, I also took the dry weight of the mycelia in liquid culture, and so I found that Fusarium proliferatum grew significantly slower in complex media broth in comparison to the Phomopsis strain. And when checking for production of conidia, I didn't find any significant difference there. Next, following Koch's postulates, I wanted to perform a pathogenicity assay. And so once again, sticking with the Fusarium proliferatum, the Phomopsis, and the Phoma strains, I inoculated ROC22 sugarcane stock. In column A, you can see this is the negative control, so just water. Column B is the Fusarium proliferatum, or column C and column D is the Phomopsis and the Phoma strains, respectively. And as you can see, the Phomopsis and the Phoma did not produce lesion sizes that were significantly different than that found in the negative control. However, the Fusarium proliferatum strain was able to cause stalk rot and therefore is the putative causal agent for the Pokkah Boeng disease. Next, I was really interested in what the relationship was of these other strains that I had isolated to the Fusarium proliferatum strain. So what interactions do they have in the field that might affect disease symptomology overall? And this is based on a couple of different works that have been done before. One of them came out of George Newcomb's lab in 2012, and the journal was in Ecosphere, in which he found a significant difference in the symptomology when the pathogen melanospora was paired with an endophyte or with a saprophyte that had been isolated in a similar field. And so column A is the Fusarium proliferatum on its own. Column B is Fusarium proliferatum with Phomopsis. Column C is Fusarium proliferatum with Phoma. Column D is the Fusarium proliferatum with Alternaria, and column E is the Fusarium proliferatum with the Epicoccum nigrum species. And so, based on the naked eye, I thought that maybe there would be a significant difference. And I used the ImageJ software to calculate the approximate area of the lesion sizes in all of my samples throughout the 3 trials. And based on analysis of variance and students' t-test, I didn't find any significant difference between the co-inoculations and the Fusarium proliferatum by itself. Now, I had the opportunity when I went to Asilomar to talk to George Newcomb's student and see what maybe she had done differently that I hadn't thought of. And so she made a couple suggestions about the timing of co-inoculations and how that might have an effect. And so it would be interesting to kind of go back and try and test a couple of different factors. But unfortunately, there's not really funding for that right now. So it is what it is, but I think that there is more here yet to be investigated. So just in summary, I isolated a total of 137 fungal strains from diseased sugarcane cultivar ROC22. I was able to narrow it down to the top 10 high cellulase producers, one of which was Fusarium proliferatum. Based on my pathogenicity assay, we saw that Fusarium proliferatum caused evident rot symptoms in sugarcane cultivar ROC22. And while the fungus didn't have the fastest growth rate, it was the most resistant to SDS stress. And the high cellulase producing strains, including strain 47 and strain 22, the Alternaria and the Epicoccum species, we found that they were not directly associated with the Fusarium proliferatum virulence based on co-inoculation studies. But we do propose that Fusarium proliferatum is the primary causal agent of Pokkah Boeng disease in the region that I was studying. So this is a really interesting experience for me. It was, I grew a lot from it. It had its ups and downs. Everything wasn't always great. I definitely struggled. I learned a lot about myself and about this new culture and how to work independently. And so it's something that I will remember for the rest of my life, and I've made some really good lifelong friends. take a quick break. Quick drink of water real quick. So the main part of my talk today is on work that I've been continuing pretty much since I started at the SHIM lab as a graduate student. And it's on the characterization of secondary metabolism regulation in Fusarium verticillioides using subnetwork module prediction. Forgetting. I'm so used to being in the class and it working. Okay. So just a quick overview. I'll be going over the motivation for my research, a little bit of background, my overall aim for the project, as well as some progress that I've made so far. If you see this picture on the right, this is a maize ear. You can see the kernels have been infected with Fusarium verticillioides. And this is really the motivation for our research in the SHIM lab. Fusarium verticillioides is a fungal maize pathogen. It causes not only ear rot, but also stalk rot. It's a heterophthalic ascomycete, meaning it can sexually reproduce, and it's ubiquitous wherever the crop is produced. The fungus is also capable of producing these mycotoxins known as fumonisins, and that's on corn ears and kernels. And the most predominant form of this fumonisin is FB1, and it has been linked to esophageal cancer in humans, as well as equine encephalomalacia. I'm always worried about that one. And so just a quick background, and it actually starts with somebody here in the room, Mr. Carlos Ortiz, Dr. Carlos Ortiz, as I should say. He was interested in looking at Mads-Box transcription factors. Now, Mads-Box are involved in the co-regulation of genes, as well as signal transduction and developmental functions. They're highly conserved across a wide range of eukaryotes and defined based on sequence similarity. And diversity of these Mads-Box can be attributed to protein dimerization. And so, like I said, Carlos was interested in these Mads-Box transcription factors, and what he wanted to do was to characterize their role specifically in secondary metabolism and sexual reproduction in Fusarium verticillioides. And so he knocked the MADS1 and the MADS2 out, and MADS1 being FMT1, the MADS2 knockout being FMT2, and he also did a double knockout mutant, which was FMT3. And what he found was that FMT1 produced 50% less FB1 than the wild type. While FMT2, the MADS2, and FMT3, the double knockout, produced 80% less FB1 than the wild type. So we knew that, or Carlos knew, that Mads-Box transcription factors were involved in secondary metabolism. And so to validate this, he used qPCR to check expression levels of polyketide synthase genes. And what he found was that expression levels decreased as time passed, as expected, with 13 of the polyketide synthase genes in FMT1, at least half of that of the wild type, 10 days post-inoculation. And so we knew that Mads-Box transcription factors are impacting the expression of PKS genes and ultimately FB1 biosynthesis. And so what we were wondering, what our aim was to find out what are the genes and pathways downstream of Mads-Box transcription factor that led to these changes. We decided to use next-generation sequencing, or RNA-Seq, to investigate downstream of Mads-Box transcription factors for proteins involved in secondary metabolism and FB1 biosynthesis in Fusarium verticillioides. And so by finding these proteins acting downstream of Mads-Box transcription factor, we wanted to functionally characterize them for their role in FB1 biosynthesis. And so in order to use next-generation sequencing, we had to find out what the best time points would be. And so using qPCR, we saw that the differential expression of genes with MADS1 and MADS2 peaked around day 4. And so we chose day 5 and day 7 to do the RNA sequencing. And so the method that Carlos used was to inoculate the MADS mutants and the wild type in myromedia. And myromedia is a synthetic culture, and it that will promote the production of FB1 biosynthesis. And so we harvested it, as I said, at days 5 and day 7 post-inoculation, extracted the RNA, and then did sequencing with 5 samples per time point per strain. Now, the expected outcomes was that it would validate what the qPCR data had said, that PKS genes and other secondary metabolite genes would be differentially regulated in the wild type versus the Mads mutants. We also expected that vegetative development genes would be differentially expressed, as well as other genes and signaling pathways under Mads box transcription factor regulation. So this is really where I came in. And so I was handed all this data, this RNA-seq data, and I didn't really know where to start. I had never dealt with this much data before. And so I took a very classical approach to looking and sorting through this RNA-seq data. And so I decided to narrow it down to the top highest differentially expressed genes in the Mads mutants versus the wild type. And of these top differentially expressed genes, I found approximately 9 genes of the FUM cluster upregulated in wild type versus FMT1. So this was like a sign that we were kind of on the right track. Amongst the top 10 most highly differentially expressed genes, I found FVEG01573 and FVEG01575. Which both encode for class 2 hydrophobins, Hyd4 and Hyd6, respectively. I also found this other gene, FVEG01574, which is an uncharacterized putative protein, and it was clustered in between these 2 Hyd genes. And it was differentially expressed, but not as highly as these Hyd4 and Hyd6. And so we thought this was a pretty good idea. Class 1 hydrophobins had been previously characterized and, in Fusarium verticillioides and found to affect Canidia morphology. However, these class 2 hydrophobins had yet to be investigated. And so we thought, okay, this is great because hydrophobins have been known to be involved in a number of different functions throughout the fungal life cycle. They've been linked to pathogenicity in Magnaportha species. They're involved in contributing to the hydrophobicity of mycelia. They're also involved in the formation of aerial hyphae and Canidia morphology. They're now used in a number of different applications, one of the most popular being beer brewing. And so obviously I was interested and thought, oh, this could be really cool. And so I knocked him out. But didn't exactly turn out the way that I was expecting. We were anticipating that there would be a difference in phenotype. However, there wasn't any difference that we had observed. No difference in hydrophobicity, as you can see in this figure over here. The pathogenicity assays showed no significant difference in lesion size between the wild type and the Hyd4 and Hyd6 mutants. And besides a reduction in fitness, we didn't see any differences in ergosterol or FB1 production either. So we hit a roadblock, right, as often happens in science. And so we were kind of stuck. We didn't really know where to go from here. And so we realized that we weren't the only ones, that this is actually somewhat common when it comes to analyzing these large data sets such as microarrays or RNA-seq. And these are the most popular outputs, I guess, or common outputs that you'll see for RNA-seq data: differential expression, you have dot plots, we classify different genes that are differentially expressed in these pie graphs, you have Venn diagrams, heat maps. And the one thing that makes all of these in common is the fact that it's taking a very single gene approach. And so what we know in biological systems is that genes often don't work at a single gene level. I mean, there are networks of genes and there's redundancy, and sometimes you knock a gene out and it's compensated by another gene. And so we wanted to take an alternative approach to analyzing RNA-seq data. Not necessarily a better approach, but an alternative approach. So we contacted our friends over in electrical and computer engineering, and we came up with this idea, well, with the help of our friends, to predict gene regulatory logic using RNA-seq data. So what do I mean by this? So you start out with transcriptomic data. This could be microarrays or RNA-seq. And then you have an inferred network structure, which has already been built by data collected by other scientists and what we know about biological systems. And when you put these together, what you can do is you can actually create subnetworks of differentially expressed genes which might be correlated to one another. And then you can analyze each subnetwork module to find how different genes are interacting with one another and what downstream effects that might have on other genes. So this was a new concept for us. We had to really kind of learn a new language and understand what the computer scientists were telling us and how they could help us. And so I'm going to go over a little bit of key concepts with this theory, and so a little terminology here. To start, all of this network building, this follows the law of organizational behavior. We see this both in biological systems and non-biological systems. And so in network measures, what you have is an undirected network. This is what is often seen for protein-protein interactions. And then you have directed networks, which is what we see for gene regulatory networks. So gene A is affected, and then it affects B downstream, and that might affect C downstream, and so on and so forth. Now, there's three types of different network models. You have the random model, in which there's pretty much an equal amount of cohesiveness amongst each member. We call these red dots nodes within the network. You also have a hierarchical network, which is completely modular. We don't see this in biological systems. Usually that's what you get completely computer generated. And so what fits both biological and non-biological systems the best is what we call a scale-free network. And just by looking at it, you could kind of see how a biological system could fit this model. And so what is our proposed network or analysis flow? Well, we start out with pretty much the same thing that we would start with using a conventional method of RNA-seq analysis. We use pre-processing. We align the RNA reads up against the genome of the fungus. We then filter and normalize based on beta tubulin. And then the same way as the conventional method, we find what we would call seed genes. And those genes are those that are the most strongly differentially expressed in the two phenotypes, so in the wild type versus the mutant, at the last time point, okay? And so normally in the conventional method, like what I had used to find the hydrophobins, that's where we would stop. We would just go on to investigate those seed genes. But instead we wanted to take a different approach, what we call the seed and extend approach. So it's a computationally efficient technique, and it's based on what we would call the log likelihood ratio, and we use a t-test statistic comparison in order to verify or improve the integrity of the networks that we hypothesize. And so I know this is a little strange, this words that we've never heard before. And so let me go ahead and explain. So you start with your seed gene, so the gene that's most highly differentially expressed in the wild type versus the mutant. At that point, we want to extend onto other genes whose expression is directly correlated with that of the seed gene, okay? And so we use the log likelihood ratio, which is pretty much where we take a gene and take its expression, which is measured in log, and then take a ratio of that of the wild type versus the mutant, okay? And every time we add on a new gene to the network, we have to improve the discriminative power, which is measured through a t-test, by at least 10%. So in this example, we'll say that every gene highlighted in green can improve the network by 10%. Those that cannot are completely removed. They're taken out. So then we extend again. Once again, those genes highlighted in green can improve the network by 10%, but these others cannot. So we're taking them out. And so you have your network, and then you can move on and go on and so forth. Now, at the same time that we're building these individual networks, we're also comparing networks, right? Because we're not just looking at one class. We're looking at 3 separate classes, that of the wild type, that of the MADS1, and that of the MADS2. In order for us to discriminate, I guess, between networks that we will accept and networks that we will not, we consider up to 3 networks whose discriminative power are within 2% of the discriminative power of the best subnetwork with the top t-score. And so in order to predict these gene-gene associations, we took 3 different types of measurements into consideration. We used Pearson's correlation, which I'm sure many of you have heard about before, and it allows you to look at the relationship between 2 genes, 2 individual genes. Okay? So it would allow us to look at A and B and the relationship between B and C. Now, the only problem with this is that if we're looking at B and C and that relationship, how do we take into account the effect that A might have on that relationship? And so we used partial correlation. And what partial correlation allows us to do is it allows us to measure the strength of association with 2 genes while removing the effect of a set of other genes. So in other words, it allows us to figure out what effect A has on B and that A has on C and remove that so we can find out the true association between B and C. We also use a type of measurement called synergy. And synergy allows us to differentiate between the different classes. So between the wild type versus the MADS1 or the wild type versus the MADS2, also at different time points or pathogenic versus vegetative. And I know this is a little bit confusing. It was confusing for me too. And so if you're really interested in how we use these measurements, this is a really good paper, Hub Discovery and Partial Correlation Graphs by Hero. I recommend you look at it. It might go a little over your head. It went way over mine, but it's a good reference point to finding out how to apply partial correlation as well as other measurements to network systems. Now, the last thing that we wanted to look at in our networks was 2 important features, one being the degree exponent. So what do I mean by that? So we want to find out what is the probability that a certain node within a network will have so many different connections or degrees, okay? And so K would represent the number of links or the number of degrees from each node, and gamma represents the degree exponent. And based on both biological and non-biological systems, we found that gamma is usually between 2 and 3. The closer it is to 2, the more highly the probability. The closer it is to 3, the lower the probability. Now, we also wanted to look at clustering coefficient. And clustering coefficient is a way for us to analyze the cohesiveness amongst the different members of nodes within a network. And so what do I mean by this? And I'll have to use the pointer so I can do it. So if you have this node here, and this node connects here, and the node also connects here, then it's a high likelihood that these 2 nodes will also connect. Okay? And so it's how many triangles will form within a subnetwork. And for hypothetical hierarchical systems, so in a perfect world, the clustering coefficient has an average of 0.6. So 60% of the time, there's a high level of cohesiveness. And so for scale-free networks, we would have to find a clustering coefficient below 0.6 because it's not a perfect world, right? So that's what is one way of testing to make sure that our subnetworks are are acceptable. Now, I know I just went over a lot of terminology. I promise this is the last slide that I will be going over as far as terminology goes. But it's a very important topic, and it's important that everybody understands it. So network robustness has to do with how stable your network is. So in most non-biological systems and biological systems, networks are very stable. They're very robust, meaning if you take one of the members out, if you take out a node, the network will still survive. It will still remain stable. This is due to redundancy and a number of other factors. And so scale-free networks in particular are considered especially robust. And it has to take a very critical member in order to completely crumble the network. And so that critical member within a network, we would refer to that as the hub. Okay? And so one goal of this project in identifying the different subnetworks is to identify the hub. What are the major critical genes that if you remove them, everything else falls apart? So we were able to come up with 4 different subnetworks based on our Mads data versus the wild type. Now, it is important to remember that these modules do not always have the largest number of genes or interactions. Remember how I was saying about seed and extend? If it doesn't improve by 10%, then we throw it out. And so just because it has a large number of genes doesn't make it better. Also, the network t-test score is always high, but when you look at individual genes, the t-test score is always low. The clustering coefficient is high, but we always want it to be below 0.6. Now, we did one last thing in order to further validate our subnetworks, is we looked at the genes within the subnetworks and then used FungiDB to look up the Go terms. And so what we wanted was for each of the members of the subnetwork to have similar Go terms. We also looked at these genes and at their orthologs and other known pathogenic fungi, such as Magnaportha, Eustalogomatus, other species of Fusarium, Cryptococcus. And through this, we identified 2 key genes, pathogenic genes of Fusarium verticillioides, FVEG0035 and FVEG07056. And like I said, we got 4 subnetworks, right? So 2 subnetworks for 35 and 2 subnetworks for 7056. And so this is pretty much where I came in, and I was given these subnetworks. And so I really wanted to try and identify what was the putative function of these genes within each subnetwork. And so I started with the key gene, the key seed genes, the putative hub genes, as we will say. What I found was that FVEG00035 encodes for a P-loop containing nucleoside triphosphate hydrolase, and 07056 encodes for a homeobox transcription factor. Now, in order to choose which of the 2 subnetworks for each gene was the best one or the most likely, I went ahead and found what using a software, I was able to find what the predicted protein domain of each gene connected directly to these 2 seed genes. And so based on the putative protein function, I narrowed it down to these 2 networks as the most likely networks. And it's important to make this point. These are not protein-protein interactions, okay? These are genes in a network whose differential expression is directly correlated, okay? And so that's a really important concept to remember about these particular networks that we've hypothesized. So like I said, we wanted to look at these seed genes and see if they really do have a role as putative hub genes. And so I went ahead and knocked them out and verified by Southern. I ended up getting 1 knockout for the FBG0035, the GTPase, and 2 knockouts for the homeobox transcription factor. So far, I've done 2 trials of growth rate assay. So far, I haven't seen any significant difference in growth rate on V8, PDA, or Myro or PDA, sorry. Although I did find that the mycelia pigment is darker in the homeobox transcription factor gene on PDA in comparison to the wild type. So it is some indication that secondary metabolism is being affected. So like I said, we want to test for network robustness. We want to find the key genes, the hub genes that affect secondary metabolism and can have an effect on other genes within their subnetwork. And so in order to verify this, I'm going to be doing some qPCR experiments to look at the expression levels to see if they're differentially expressed in my new knockout mutants in comparison to the wild type. I also want to characterize the role of ergosterol and FB1 production in the knockout mutants versus the wild type using high-performance liquid chromatography. So just a quick summary. Mads transcription factors are associated with secondary metabolism of Fusarium verticillioides. We use next-generation sequencing, or RNA-Seq, in order to discover novel secondary metabolism regulatory genes downstream of Mads transcription factor. Using the conventional approach, I identified 2 highly differentially expressed genes, including HYDE4 and HYDE6. I knocked them out, but unfortunately, we didn't get the anticipated phenotypic results. So instead, we went with this network-based approach in order to identify correlated genes in the regulatory network. After building our subnetworks using this approach, we identified 2 seed genes, putative hub genes, 00035 and 07056. And I'm now characterizing their role in fumonisin and ergosterol production, as well as the effect that they have on other genes within their subnetwork. So just to finish, I'd really like to thank the Shim lab. Dr. Wambo Shim has been a very amazing mentor to me. Juan, I wouldn't be here without her help and guidance. Jimmy Kim, he's the graduate student who has been doing all of our statistical analysis for us. He's over in electrical and computer engineering. Unfortunately, he couldn't be here today. He just had a little girl, so he's busy being a dad. I'd like to thank Carlos Ortiz for initiating the Madsbox project and handing it off to me. And then, of course, Wenying Yu, she was a visiting graduate, or a visiting scholar, I should say. She's a professor now at FAFU, and she was a huge help when I was there in FAFU, as well as when she was visiting here in the United States. I'd also like to thank my advisory committee, Dr. Charles Kennerly and Dr. Daniel Ebley, and the key collaborators that have helped us with this project. Can't forget the funding source, the USDA, the Genomic Seed Grant Program. I'd also like to thank the College of Agriculture for giving me the Sloan Diversity Fellowship, which has helped pay for my master's program so far, as well as the Department of Plant Pathology and Microbiology. Thank you. Yes. Yeah, we haven't really looked at that. I mean, it is one of the limitations of this approach is that it is hypothetical, and without doing the actual bench work and testing the biological system in the lab is really the only way that we can validate that. So that's really where I've come in as a researcher. I didn't predict these subnetworks. This was done by our computer engineering friend, and so I'm doing the legwork, so to speak, to provide evidence to support these hypothetical networks. Yes? So what's interesting is that those hub genes are not actually the most strongly differentially expressed. The other interesting thing is that the hydrophobin genes that I had originally identified that were highly differentially expressed will not form networks. Yeah. Yes. So I know I mentioned the discriminative power, and it's based on a t-test. And when I say t-test, this isn't the standard student's t-test that we do here in biology or in plant path. This is a t-test that they use in statistics and computer science. And I wish I could better explain it to you, but it's been even kind of hard for me to fully understand. Dr. Shim, do you have any additional comments on that? Yeah. Yeah. Mm-hmm.
They make the network stronger.
Yeah.
And so
That's a perfect explanation. Thank you, Dr. Wilkinson. No, that's perfect. Better than I explained it, really. But yeah, what I mean by it's not a standard t-test is that when our computer scientist guy came in and we were asking him, you know, like, what is the significance, you know, what is your confidence level and everything like that, and he was kind of like, oh, well, it's like arbitrary, and so it's different in what we would normally classify as a student's t-test, and so it's kind of hard for me to explain it, but I do have several papers that if you're interested in reading, then I can definitely recommend them to you. Yeah. Yeah. Mhm. What do you mean exactly? I mean, do these have orthologs in other fungi? Well, I'm sure. I mean, protein kinase domain. I mean, I haven't looked at that. It's a good question, but I hadn't looked that far into it. I really kind of just wanted to look directly at them. Does this make somewhat a biological sense? So, in other words, could these genes be differentially expressed in correlation to my seed gene? And so that's how I narrowed it down to these two networks. So I really haven't looked that far into it, but that's a good question. Yes. Oh, sorry. I like that you asked that. I'm sorry I didn't mention it before. I should have. So red indicates upregulation in wild type versus MADS mutant. Blue would be downregulation. So these don't necessarily have to be correlated in both going up or both going down. It could be an inverse correlation that's occurring. I haven't yet, and part of that is because the limitation with greenhouses. So I haven't gotten the opportunity to do that yet. I have put them on kernel. So far, I'm not seeing a significant difference, but I can't speak to that because I need to do more trials. Yeah. I mean, it's much harder. I mean, it's much harder to grow mature maize inside growth chambers. They're much happier outside. So even inside the greenhouse has been challenging for us. Both the MADS1 mutant and the MADS2 mutant versus the wild type. So we're not just looking between wild type and MADS1, but we want to make sure that it's happening in both mutants in comparison to the wild type. True, yes. Mating was also impaired in the mutants, but we've chosen not to go down that path. We're really focused on secondary metabolism. But if you can remember in the earlier slides, Carlos validated the regulation of secondary metabolite genes using qPCR, in which he showed that polyketide synthase genes were differentially regulated. Yeah, and then the fume genes were also differentially expressed. So
hypothesis 5 and 7.
well, these are subnetworks, so it's important to realize that in the biological world, right, it's a huge network, and then these are possibly, yes. Not a direct interaction, but yeah. Yes. Yes. we haven't looked into that. It would be an
Thank you very much.
Thank you.
Sampling. It's looking at patterns of relationship, and that's why